Thanks very much for having me. It's a pleasure to be here. And I try to come up with an entertaining title, but this is really going to be a talk about diarrhea. To introduce the topic, I'm going to take us, I'm going to take the approach to look at the eyes of a medical student, because that forces you to take a step back and look at the big picture. So when I teach medical students, I tell them the most important thing you have to remember about diarrhea is that it comes in two flavors that are represented by a patient that has no fever or low-grade fever and no white blood cells in the stool sample, versus another patient that would present with fever, and if you would take a stool sample, you would find neutrophils. And then as a good-looking physician, you would diagnose secretory diarrhea versus inflammatory diarrhea, which is important because the organisms causing this are different. So on the secretory end of things, we have Vibrio cholera, enterotoxigenic E. coli, and the two organisms we just heard about, the AE pathogens. And on the inflammatory end of things, you have Campylobacter shigella and non-typhoidal Salmonella, which is the topic of our talk today. So I'm a PhD. I was brought up learning all these organisms one by one, and each one has lots of virulence factors. And then you grow up and start teaching medical students, and you realize there's only two ways for the host to respond to these different insults. And that tells you an important concept, and that is that host responses are always characteristic of groups of pathogens. And that means this group has something in common. This distinguishes it from this group. And what that is important because that determines the patient presentation. So that's the first thing the medical student should know about them. The property these organisms here have in common is that they are all invasive. They enter the epithelium. They're detected by the innate immune system, which causes a vigorous inflammatory response. Cytokines are produced. Some of them, like interferon gamma, reach the bloodstream and enter the brain, where they change the temperature set point, so you have fever. Other cytokines, called chemokines, they attract inflammatory cells, in this case neutrophils, and these neutrophils migrate into the intestinal lumen in an attempt to protect the epithelium in large numbers, and you find them in the stool sample, and you will see during this talk that property will become very important. Now, in contrast, this group of organisms here is a group of luminal pathogens that are not invasive, and therefore the inflammation they cause is much milder and doesn't dominate the patient presentation. Not to say that these cause no inflammation, so they do cause some inflammation, and you can measure cytokines, but it's just compared to these organisms, it's much less severe because they stay in the lumen and don't invade the tissue. So medical students usually like this because it helps them understand the patient presentation. We get evaluated every year, and so a couple of years ago, one of the students commented, "I like diarrhea now." Just the kind of compliments I get, I guess. But I assume most of you are not MDs, but you're PhDs. So you don't really care about the patient that the medical students want to understand. You care more about these organisms. And so if you want to understand the pathogen, you have to look at this from a different point of view. And I think it was best expressed by a Nobel laureate from France. What the pathogen is really out for doing is to replicate. And for pathogens that are persisting in a host reservoir, what that means is that the pathogen has to transmit from a naive host, from an infected host to a naive host, right? So it's all driven by transmission. So if you want to understand a pathogen like Salmonella, you have to understand how it uses its virulence factors to promote its transmission, right? And so this is what I'm going to try to do today. So I'm going to start with the virulence factors. We didn't have a talk yet introducing this formally. So Salmonella has two major virulence factors, which are both type 3 secretion systems. One required for invasion, the second one required for survival in tissue. And the common model to study this is the mouse model. Susceptible mice are typically used that succumb to infection between 6 and 10 days. And the dirty little secret of Salmonella pathogenesis is that the mouse develops a very different disease than the human. The mouse gets a bacteremia with very little intestinal inflammation, whereas the human gets a localized gastroenteritis. So we don't get bacteremia, at least if you're immunocompromised, when you have non-typhoidal Salmonella, which is Salmonella typhimurium, the organism I'm studying. So when I was in Texas, I was establishing a different model in the calf, and calves are animals that develop the exact same disease as the human, gastroenteritis, localized infection, localized to the terminal ileum and colon. And what you see here is the lumen of the small intestine, the terminal ileum, 2 days after an oral infection with Salmonella. And you see this layer of pus, essentially. This is a pseudomembrane composed of fibrin, neutrophils, and necrotic tissue. So neutrophils come out in large numbers. And when you can peel off this membrane, you see this reddened mucosa, which is due to massive inflammation. So this is a lesion that covers a large area, 2 to 3 meters of the terminal ileum and 1 or 2 meters of the colon, which look like that when you have Salmonella. So it's a pretty bad infection. Virulence factors are important for this host response. So when you inactivate the type 3 secretion system for tissue survival, the inflammation is reduced. And if you inactivate the type 3 secretion system for invasion, there is no more inflammation and the calves don't get diarrhea. Now, this host response is quite good in clearing the organisms from the tissue. So neutrophils are very good killers of Salmonella. So within 7 to 10 days, bacteria are cleared from the tissue as inflammation subsides. So I told you what you need to understand if you want to understand a pathogen is to know how it uses its virulence factors to promote its transmission. Now, Salmonella uses its virulence factor to cause a host response that kills it. So that's paradox. This doesn't really seem to be intuitive that this is something the organism does in order to stay on this planet. But if you look at this situation a little closer, you see there's 2 populations of Salmonella. One population is the one that enters the tissue, so the invading organisms shown here in a immunohistochemistry stain. So these organisms are the ones that trigger all the havoc, all this inflammation, and they're the ones that get killed by the host response. But you see there's a second population in the lumen that does very well. But Finley initially showed for Citrobacter that during inflammation, Citrobacter can expand in the lumen, and that was shown also by Bärbel-Stecher later for Salmonella. So Salmonella is doing very well in this environment and expands. And then Denise Monarch had shown that this is actually required for transmission by the fecal oral route. So Salmonella uses its virulence factors to transmit this way. But how this worked exactly wasn't quite clear. And so we got interested in this. And we thought we could maybe get at this initially from an evolutionary point of view, because there's a close relative of Salmonella, which is E. coli, a group of commensals, essentially, even though it produced many pathogens over time. Most of you have an E. coli in you, which is a commensal at the time, whereas all Salmonella are considered pathogens. And they diverged quite some time ago. My grad students like to point out around the time I did my PhD. And shortly after that event, they acquired these virulence factors on pathogenicity islands, these 2 types of secretion systems that enabled the pathogen to cause this massive inflammation, which could be seen as an event of niche creation, so that the Salmonella colonizes the inflamed intestine, whereas the microbiota colonizes the non-inflamed intestine. And to exploit this new niche, you would have to propose that Salmonella acquired genes at around the same time that allow it to take advantage of this niche. So we were thinking what we are looking for are genes that are conserved in Salmonella and that allow Salmonella to outgrow any other bug. And there was such a thing known from the way we isolate Salmonella in the clinical laboratory. So when you have your infected burger sample, you put it in enrichment broth, and the next day you go on selective agar, and then you pick a colony for serotyping. And that enrichment broth is tetracyanate broth or some derivative thereof, which was developed in 1923, apparently by a French group. I guess from the title. The broth works because tetracyanate can be used by Salmonella as an electron acceptor for anaerobic respiration. So instead of oxygen, they can use tetocyanate, reduce it to thiosulfate. The genes for that respiration were acquired early on in the evolution of Salmonella, so they were present in all Salmonellas. So we have something which is conserved, which allows Salmonella to outgrow other bugs that look promising, but we didn't know where the tetocyanate was coming from. Sebastian Winter in the lab was taking on the project, and he got interested in intestinal gases and in sulfur metabolism. And so one gas in particular, which hydrogen sulfide, is one of the gases that makes excrement smell interesting. But it's also very toxic, and so the toxicity is so high that the apical surface of epithelial cells in the colon, they encode these sulfide oxidases that detoxify hydrogen sulfide to thiosulfate. Thiosulfate is the end product of tetocyanate respiration, but when we actually looked at the bottle of tetocyanate broth, we noticed that tetocyanate broth actually doesn't contain tetocyanate, it contains thiosulfate. Reason being that tetocyanate is fairly unstable, so if you would put tetocyanate in this broth, it would have a very short shelf life. So the company gives you a second bottle, which is a bottle of iodine, and you have to add that iodine to the tetocyanate broth just before you use it. Iodine is a strong oxidant, and it oxidizes thiosulfate to tetocyanate, and now you have your tetocyanate broth. So when we saw that, it kind of clicked. We thought, well, that's what happens during Salmonella infection. I told you there's neutrophils coming out. This is how we distinguish inflammatory diarrhea from secretory diarrhea. So the neutrophils in the lumen, which is here, come out in large numbers, and they undergo a respiratory burst. They generate a lot of radicals, and these radicals then replace the iodine in this reaction, and you get tetocyanate produced by this way. So we wanted to test this, and in order to have a model, the mouse model I told you is a little problematic. Mice don't get very much inflammation when they're susceptible to Salmonella before they actually become moribund. But Wolf Hart had developed a nice model where mice are treated with streptomycin one day before infection, and then in that setting, mice become much more pronounced inflammation, and you see actually neutrophils migrating into the lumen. So we decided to use that model to ask whether tetocyanate respiration is important. So we infected mice, and what you see here is the cecum of a healthy animal, mock-infected animal, the mucosa, submucosa muscularis. At the same magnification, the cecum 4 days after infection with Salmonella, where you see this massive thickening of the mucosa due to neutrophil influx, and edema in the submucosa, so massive inflammation. And then, you take the cecal contents and you give it to your metabolomics core facility to measure tetocyanate. And they tried, and here's a little error bar. So there was no tetocyanate in the gut. So I thought, that kills this project dead. But Sebastian wanted to do one more thing. He said, well, maybe tetocyanate is limiting and Salmonella consumes all of it. So we should repeat this experiment with a Salmonella strain that cannot consume tetocyanate. And I wouldn't be talking about this if it hadn't worked. So when you inactivate the tetocyanate reductase of Salmonella, you still have massive inflammation, but now you can measure tetocyanate. So it's all consumed by Salmonella. It's limiting growth, and the effect of it is that you have an expansion of Salmonella. So this is Salmonella in red, and in order to expand to a considerable fraction of the microbiota, you need tetocyanate respiration, and you also, of course, need the virulence factor. So if you don't have inflammation because you inactivate the two types of secretion systems, you also cannot expand. So this is kind of how we got into this. And the reason I'm bringing this up is because when we started, you always plot these microbiota data on this pie graphs, which show you basically 2 logs of growth, but it's really just the tip of the iceberg. Underneath, there's a lot of data hiding. So here, your tip of the iceberg is this 25-fold increase you see here, which is driven by inflammation and virulence factors. But when we did this assay, we treated mice with streptomycin, and we just heard about a couple talks ago that this removes colonization resistance, which is something you see, of course, also in this setting. So this antibiotic, in the absence of antibiotic treatment, you have much lower numbers of Salmonella. This is totally independent of virulence factors because you see it with the avirulent mutant that does not have the secretion systems. And because it's independent of virulence factors, you also see it in E. coli, and it's a much larger effect than what we saw up here, so it's a 1,000-fold effect. And we became interested in what the basis for this was. So initially, we did some work in E. coli. This was Alana's work, who noticed that there was streptomycin actually induced expression of NOS2, which is the gene encoding inducible nitric oxide synthase, the enzyme producing nitric oxide. And nitric oxide was interesting to us because it can be converted in the gut to nitrate. And nitrate is a very good electron acceptor, the favorite electron acceptor of Enterobacteriaceae under anaerobic conditions. E. coli and Salmonella have 3 nitrate reductases encoded by these genes. And so we compared the fitness of an E. coli wild type with a mutant that is not able to respire nitrate in a mouse model. And so you see that when you infect a mouse with a 1-to-1 ratio of these strains, you recover a 1-to-1 ratio in the absence of streptomycin, suggesting there's no nitrate respiration going on. But if you treat with streptomycin, you see a benefit of nitrate respiration, a fitness advantage. But it was only about 10-fold. And what you saw with colonization resistance was a 1,000-fold effect. So there was something, a big piece missing from the puzzle, and we were, for a long time, we didn't really know what that was. And finally, Fabian in the lab was managing to make some headway, and that's because he read up on the physiology of the colon. So the colon is really functioning in fluid absorption. And in order to absorb fluid, what the epithelium does, it pumps electrolytes through the membrane, so generating this osmotic gradient, and then water follows passively through the tight junctions. So this pumping requires energy because there's an ATPase in the basolateral membrane which does that. And that ATP driving this process comes from respiring butyrate to CO2. So butyrate is the energy source of colonocytes. That butyrate comes from the fermentation of complex carbohydrates in the large intestine. The Bacteries are the ones usually breaking down these complex carbohydrates, but then the fermentation products produced are converted into butyrate by one group in particular, which is the Clostridia. And so this butyrate is then respired, and apparently the amount of oxygen consumed in this process is so large that the epithelium in the colon becomes hypoxic, which is less than 1% oxygen. So normal tissue oxygenation is between 3 and 10%. So what Fabian found, and what we seen also before, is when you, in an earlier talk in the morning session by Karina, is that when you treat mice with streptomycin, Clostridia get depleted dramatically. So one day after streptomycin treatment, we see at least a 6-log drop of Clostridia. And I told you Clostridia are the main producers of butyrate. So when you measure butyrate levels, they plummet by about 4 logs. And this is what we measure in germ-free mice. So there's no butyrate left. And if there's no butyrate left, this metabolism can no longer run. And what the epithelium does, it switches its metabolism to this fermentative metabolism where you ferment glucose to lactate. And that process does not consume any oxygen, so there's oxygen levels go up in the epithelium. And since oxygen diffuses freely over membranes, Fabian postulated that this process somehow would increase oxygen availability in the lumen, and perhaps oxygen was our missing piece. So we wanted to test this, and the enzymes relevant for this were these 3 oxidases. The BO oxidase is mainly functioning under high oxidation in atmospheric oxygen conditions. So we focused on these 2 here, the BD oxidase, which is working under tissue oxygenation, and then the BD2 oxidase, which wasn't very well characterized, was just identified by genome sequencing. So the physiological role of this enzyme was not known. So we first inactivated the BD oxidase, and when you compare the growth in vitro between the wild type and the strain lacking this enzyme, you see that in tissue oxygenation at 8% oxygen, you see a nice advantage of this enzyme. But if you go below that to 0.8% oxygen, there's no advantage. suggesting that this would be required for growth in tissue. So when you infect mice IP to circumvent the gut and then recover bacteria from the spleen, you see that this enzyme here, the SUD-AB, BD oxidase, is generating a nice fitness advantage in the spleen and tissue, whereas this other enzyme had no role during growth in tissue. However, the other enzyme was interesting because it conferred a benefit at lower oxygen levels. So at 8% tissue oxygenation, there's no benefit of this second enzyme, the BD2 oxidase, encoded by 6A. But at 0.8%, you see an advantage, and this is dependent on oxygen because in the anaerobe chamber, there's no benefit. And so then the question was, does it confer a benefit in the gut? And you see that BD, this enzyme up here that confers no benefit in the gut, but BD2 is the enzyme that Salmonella uses to respire oxygen in the gut. And you only see this after streptomycin treatment. So when you leave the streptomycin out of the assay, you see no longer a benefit. So I told you in the beginning that nitrate respiration confers a 10-fold fitness advantage in E. coli. It does the same in Salmonella. Oxygen respiration is about a 10-fold fitness advantage, but we wanted to understand a 1,000-fold advantage, right? So we thought, well, if you combine those two mutations, what happens? And what did happen was that now we have our 1,000-fold effect. So this is colonization resistance. It's a respiratory niche. And just to show you this hypoxia, so there's a stain developed for the cancer field because the center of tumors are hypoxic, so there's pimonidazole injected in animals, and that reacts only to tissue that has hypoxic conditions. And in the colon, when you do this pimonidazole staining, you see the surface of the colon here is lighting up like a Christmas tree with this hypoxia stain. So the colon is hypoxic. You treat with streptomycin, the hypoxia is gone. When the mice are infected with a mixture of wild type and BD2 oxidase mutant, without streptomycin in this condition, there's no benefit. But as soon as you have the antibiotic, you see a nice contribution of aerobic respiration to fitness. And you can then ask, is this really due to butyrate? And to do this, you cannot just give mice butyrate because the butyrate gets absorbed in the small intestine. So you have to give them tributyrin, which is butter flavoring. And tributyrin is an ester of 3 butyrates and a glycerol, and that passes through the small intestine and then is cleaved in the large intestine and elevates butyrate levels. And it restores hypoxia, and there's no more oxygen respiration going on. All right, so this is a colonization resistance. We have normally this respiratory metabolism of epithelial cells after antibiotic treatment, and depletion of butyrate epithelium switches the metabolism. This is a common switch in the body. So this is the switch between M1 and M2 macrophages, for instance. And if you're an immunologist, you know that one of the markers of this metabolic program of M1 macrophages is inos expression. And apparently that also happens in the epithelium. So you get nitrate produced, oxygen is coming out, and you have this respiratory niche where facultative anaerobes can expand into. So I was happy with this, but Fabian wasn't quite satisfied because he said this cannot possibly be the physiological role of this enzyme here, because antibiotics are around for less than 100 years, and Salmonella is around for millions. So there has to be another reason that Salmonella has this gene, and we didn't see a phenotype without strep treatment. And we thought perhaps that is because we used these susceptible mice that die really fast, and we can only look at early time points at 4 days, where in the absence of strep treatment, we don't see a phenotype. So he switched to a different model, genetically resistant mice, where we repeated this experiment, competing the wild type with the respiration deficient mutant. The results are shown a little different here. So the wild type is in black, the mutant is in blue, and the red dots connect bacteria recovered from the same animal. So you see they all recovered at 1-to-1 at 5 days after infection without antibiotic treatment. So that's what you see without antibiotic treatment in susceptible mice. But now you can let this experiment go longer, and you see that there's a very nice phenotype developing over time, by day 28, the mutant is cleared from the animals. The wild type persists in very high numbers. So there's a very strong effect of respiration in the absence of antibiotic treatment. How does that work? So Salmonella invades, survives in tissue. That triggers this cytokine storm, which you can measure by looking at lipocalin in the feces. When you do this, you see that it takes about 10 days in these resistant mice to develop full-fledged inflammation, and that persists then for a week or so. You have massive cytokine production for neutrophil chemoattraction, and then, of course, neutrophils come and migrate into the lumen, which you see here. And Brad Finney's lab had shown before that neutrophil transmigration into the lumen leads actually to depletion of Clostridia. And this is what we see too. So Clostridia are in red. When you see during Salmonella infection, when the neutrophils come out, Clostridia are depleted both 10 and 17 days after infection. And you see this expansion of facultative anaerobes, both bacilli and in yellow is Salmonella. Since Clostridia are depleted, what happens to butyrate? So when you infect mice with Salmonella, butyrate levels drop in this setting by about 2 logs. And so then you can ask what happens to the hypoxia. So normally the colonic surface is hypoxic, as I showed you before. When you infect with Salmonella, hypoxia disappears, the epithelium becomes fully oxygenated. And then you can ask, is that really due to Clostridia depletion, right? So we added back Clostridia in 3 different times. It's a community of 17 human Clostridia isolates from Kinya Honda in Japan we used for this purpose. And when you do that, butyrate levels increase and hypoxia returns to normal levels. So then, you can ask what happened to these indicator strains, one which can respire oxygen, one which cannot. So if you look at wild type situation where we have no more hypoxia, you see a phenotype for oxygen respiration, but if you treat with Clostridia, this benefit is crushed. So that brings me to my last slide. It's the Salmonella pathogenesis in a nutshell. So Salmonella is a pathogen that invades the epithelium and then survives in tissue, which triggers this massive cytokine storm. Neutrophils come and actually are quite good in killing Salmonella. So going in here is kind of a dead end, and the invading population is really more like suicide bombers. They don't benefit themselves, but their comrades in the lumen do, because neutrophils migrate into the lumen. They deplete Clostridia, which leads to a drop in the butyrate concentration. The epithelium, therefore, has to change its metabolism so that now we're fermenting glucose to lactate, and the oxygenation in the epithelium increases, and oxygen diffuses out at a higher rate. I showed you in the beginning that endogenous sulfur compounds are oxidized into tetrathionine, which is an allantoin acceptor. And in case of Salmonella infection now, the nitrate comes mostly from neutrophils because they make more than epithelial cells. And Salmonella, I didn't have time to show you, uses energy taxes to find pockets in the intestine where it finds these electron acceptors, and then respires these electron acceptors to expand in the lumen. And also didn't have time to show you, but it's in this paper, this respiratory metabolism is required for transmission by the fecal all around. Through the need to transmit, this entire metabolism and the virulence factors are under selection. So Salmonella will always be a pathogen because that's what it needs to be to transmit. It's also interesting. So I told you that you need to understand how Salmonella uses its virulence factors to further its transmission. So what Salmonella does, it uses its virulence factors to use the host lung to breathe in the gut, which is a really nice trick to make a living. Okay, and perhaps the take-home message for microbiota people. If you see a bloom of Enterobacteriaceae or facultative anaerobes in the gut, the first question you should ask yourself is, where do the electron acceptor come from? Right? And I don't see many people asking this question, but respiration is so much superior to fermentation that it's just almost a no-brainer to ask this question. So when you have respiration, you get 36 ATP out of a glucose versus 3 from fermentation. But more importantly, with respiration, you can eat all the fermentation products that the microbiota produce, so you can sidestep the competition. Right, so respiration is such a big event that when respiration electron acceptors become available, this is really the main factor that's going to change the composition of the microbiota and leads to your dysbiosis. And with that, I would like to thank NIH and USDA for generous funding. Thanks.